My name is Kendall, and I'm a graduate student in the Wildland Resources Department. And it's my pleasure today to introduce our speaker, Dr. Anthony Darrazet-Nardi. He comes to us from the University of Texas at El Paso, where he is an assistant professor in the Biological Sciences Department. He completed his PhD at the University of Colorado at Boulder, where he studied with Bill Bowman. And since 2010, he's conducted postdoctoral research with Dr. Mike Weintraub at University of Toledo, and Dr. Sasha Reed and Dr. Jane Belknap at the Canyonlands Research Station in Moab. Anthony has studied biogeochemical processes in a variety of environments, including mountain, Arctic, and desert ecosystems. And today he's going to share some of his findings with us. So, welcome.
Okay, all right, great. Well, thank you, Kendall. Once again, like I said yesterday, I've had a really great visit here. It's been really nice chatting with all of you. And by the way, Kendall's been a fantastic host, very attentive, and made my trip go very smoothly. So thanks to her for that. Okay, so today I'm excited to share. Yesterday I did, kind of like a more broad theoretical thing connecting, various types of research that I did. But today I want to talk about a specific project that I worked on as part of my second postdoc that I did, as Kendall mentioned, at the U.S. Geological Survey in Moab, Utah. And I was working on these soil crusts. So I'll tell you guys a little bit about this pretty cool climate change experiment that has been set up there for a while. Okay, before I do that, I just wanted to put in a quick plug for UTEP and our Biological Sciences Department there. And I know most people don't know too much about UTEP. I didn't know too much about UTEP before I took a job there and moved to El Paso. But El Paso is actually a really interesting and fun place to live that I've enjoyed quite a bit. We have a growing department. We have a brand new ecology PhD program, which we're just starting to graduate students from. So that's pretty exciting. And actually, interesting UTEP news today, our president, we have this really fantastic college president who was selected as one of Time's 100 most influential people just this morning. So yeah, it's an interesting place, and we kind of, UTEP's unique in being this university that is majority Mexican-American. It's really the only research university that's like that. So, it's a fun part of the mission, and I've enjoyed starting to build my program there over the last year or so. Okay, so yeah, okay. The project I'm going to talk about today, I was working on with Sasha Reed and Jane Belknap were my two postdoc advisors at USGS. Both really special and fantastic scientists, cool people to work with. I mean, Jane is the world expert on biological soil crusts, and Sasha Reed was actually a graduate student with me at the University of Colorado. But she has been tremendously successful since then in building a program here in Moab. Working with Sasha on some of these same, working with Jane on some of these same soil crust questions. And then Ed Grote is a permanent tech who works at USGS, and he's responsible for all the technical stuff having to do with the equipment that I'll be talking about today, and these automated CO2 chambers. It's a huge field operation to do this, and so of course there's a lot of field techs and collaborators and folks who participated in that as well. Funding for this project, this is a DOE project funded by their Office of Science, and also USGS provided some support for it as well. Okay, so a little bit of background information. Why might we care about carbon in desert and other dryland ecosystems? I was just telling you guys yesterday, there's no carbon in the desert soils, it's not important. But like I was also saying, there's something like 40% of the terrestrial surface is dryland, so it's a huge area, and any small fluctuations in those dryland areas can potentially have big impacts. And there's some evidence that these carbon pools that are in these soils get exchanged with the atmosphere relatively readily. And here's one example. There's been a few pretty cool papers coming out recently that have shown the importance of carbon in semi-arid and arid systems. So this is showing, I think these guys looked at above ground carbon or I think this ABC is above ground biomass carbon. They looked at these trends globally over a period of, I think, 20 years. And they saw some interesting things. So tropical forests are losing a ton of carbon because we keep cutting them down. I mean, something like it used to be 10% of the Earth was tropical forest. Now it's like 5%. So we've cut a lot of those down, and it releases a lot of carbon when we do that. At the same time, boreal and temperate forests, a lot of them are aggrading and gaining carbon. And then also after that, we have some of these dry areas where you wouldn't think there'd be a lot of action, but actually the numbers are not that insubstantial. And this is just the above ground part of the story, all right? Not even the soil carbon stuff. Here's one figure I showed yesterday, and I think it kind of emphasizes the importance of having this soil carbon in these dryland systems. As you know, with climate change, if dryland systems start to get even hotter and drier than they already are, we might expect that the amount of carbon in these soils might decline, as suggested by this correlation of carbon with the aridity index over time. Drier, more arid systems tend to have less carbon in them. Some of these systems might be heading in that direction if they warm up even further than they already have been. Yeah, and I think there's some evidence that global change factors could be altering land cover, which may also have an impact on the carbon storage in these systems. There was a pretty good paper that was released by some folks at UNM recently, by this author here, showing that as you transition from grassland to shrubland, there can be some substantial impacts in the carbon balance in those systems. And of course, that transition is something that we've seen happen in quite a few places. Interestingly, as you go to shrubland, these findings suggest that you might actually be storing more carbon. Because for whatever reason, they're seeing less respiration in those shrub systems than in the grass systems, despite similar productivity between them. So yeah, that was kind of their findings from that. Point is, these pools of carbon in drylands can change pretty easily, and they might be pretty important even on a global scale. Here's perhaps the most interesting thing about semi-arid carbon. This paper came out a couple years ago, and I think there was another one that basically showed some really similar findings. These guys showed that the year-to-year variation in the total carbon balance, I mean, every year, right, we see carbon go up, due to vegetation decomposition in the northern hemisphere, and then come back down during the growing season as the vegetation sucks it back. It's the Keeling curve, it just goes up. But the year-to-year anomalies in that trend appear to be strongly driven by semi-arid ecosystems in particular. And in this particular set of years that they were looking at, they saw what they said was 6 consecutive seasons of increased precipitation in Australia. It locked up a whole bunch of carbon in that ecosystem. So there's a number of good reasons to be looking at carbon in semi-arid ecosystems, even though the total stocks of carbon in those soils are not as big as they are in other places. And actually, I did a little estimate of that. So yeah, dryland, like I said, 41% of the terrestrial surface. My best estimate, and I have a little blog post about this, about sort of how I calculated this, but something like 13% of the total soil carbon is in these drylands. So it's disproportionately low, but it still might be a fairly active pool. Okay. Jane Belknap set up this climate change experiment, I think around 2005. So it's been around for quite a while. It's still running. And her main question, her overarching interest is in the relevance of biological soil crusts to ecosystems. And she was wondering, how will climate change potentially affect biological soil crusts, which are a very common part of all these semi-arid and arid ecosystems that I was mentioning. So here's a picture of her experiment that she set up here. This is in the Castle Valley. Let's see, I think I have a map. Yeah. This is in the, you guys know where Moab is here in Utah. But this is sort of in the Castle Valley area just outside of Moab as you drive up the river there. Okay. All right, so a quick word about biological soil crusts. And again, I know with this audience, you guys mostly know what this is. But for anyone who doesn't, and just as a good sort of review, so Jane's definition from her book on soil crusts is that they are these consortia of a bunch of different organisms that have mutualistic relationships. So we have there's an abiotic component, the soil particles that are in there. cyanobacteria that are photosynthesizing and potentially fixing nitrogen. Things like you can get algae, you can get microfungi, or many lineages of fungi that have been documented in these. Lichen, and then the more well-developed ones, you get lichens and bryophytes, mostly mosses that'll crop up on these soils. So I threw in a couple of cool crust pictures here. And anyone who's traveled in this area, and these landscapes are pretty familiar. These big crusts, I took this one, at Natural Bridges National Monument. It's a relatively high elevation park, gets nice cold winters, and it really helps those crusts, the freeze thaw in the winter helps them grow up into those bigger, crustier formations that you see there. And you can see that in some areas it's a really dominant part of the landscape. It's really covering a huge amount. But even if you don't see those big, obvious crusts, there can still be quite a lot of crust organisms in a lot of systems. Here's a picture of another cool one. This one has a couple of different kinds of lichens on it. This one is in a pretty calcareous soil that's in Arches National Park. There's an area that has a bunch of these really interestingly colored lichens growing on the crust there. I just threw this one in. So this is like a close-up of one of those crusts. And you can see how big they get. Sometimes they get up to 10 centimeters in height. They can be pretty substantial in that area. And then here's a picture. A lot of times when it rains, like around the Moab area, the soil just turns green. It's incredible. And here's an example of some of the mosses on this crust. The mosses are super cool. They can dry out completely and then rehydrate themselves when it gets wet, which is pretty unusual for a plant to be able to do that. So that's one capability that they have. Okay, so the experiment that was set up involved basically two treatments. There was an infrared warming treatment that's set to a target of 2 degrees C, I believe, on the soil surface or just below the surface. I can't remember where their measurement for that is. But the idea is that in the future, if these systems get even warmer than they are now, what's going to happen to the crusts. There's also a number of different watering experiments or watering treatments that they have put in place at these within these plots. They started one set of them originally that was a smaller amount of water. They got some results from that, and then there's a few other variations on that watering treatment. Well, one of the first things they noticed was that when they used this watering treatment where I think they watered something like 10 times over the course of the summer, and they gave a relatively small amount of water to the crusts would wet up, the mosses and things would rehydrate, they'd turn on, and then they'd dry out pretty quickly. And it turned out that the crust really didn't like this particular watering treatment. And what you can see here, this is from one of Sasha's papers. We have these treatments. Here's the control in green, warming in red, and then here's the watering, and then the combined treatment here. And you can see this is the moss species that grows on those pretty substantial crusts that are at that site. And the ones that were watered just got totally hammered. Their percent cover went way down, and they were not able to survive this. And I think, in a way, the watering treatment they did might not be exactly what would happen with the future climate change, but it really demonstrates how sensitive some of these organisms are, even to relatively minor physiological impacts to them over the course of the experiment. So that was the first big finding that these guys saw here. And then one of the other, percent cover of what's going on with the crust and the plants was one of the key variables. The other thing that they wanted to look at, of course, was carbon, which is why I set up the carbon question at the beginning. And they set up this really nice system. This was mostly Ed's work of these automated soil chambers. And what these things do is basically just a chamber system that passively builds up, or it monitors the CO2 over a set measurement period, which is about 4 minutes. And these chamber tops close down on the chamber like this once per hour. They have 20 of these up there, 5 of them in each of the treatments. And for these data, I've only analyzed the control and the warming treatment. By the time they had these set up, a lot of the mosses had completely died, so I didn't focus on that portion of it. Although it would be interesting to go back and look at it, and it's something that we want to do. But it's a lot of work to go through these data because they measure once per hour. You have 20 of them. They have to be quality checked and all this thing. So it's a pretty big process to go through working with all these. OK, so here's some of the data that we get out of these chambers. And I really, I find it to be a fascinating data set because you can just kind of really see these crust-covered soils sort of breathing in and out on an hourly basis. And you can kind of figure out, what are they doing compared to the, driven by the various conditions that they are exposed to. Okay. And so what we're looking at here, this is our, on this upper panel here, this is the, what we're calling the net soil exchange of carbon. Okay, and the reason and one more thing I should point out here, we have clear lids on these chambers, so when the mosses or when the crusts are photosynthesizing, you can see carbon uptake at that time. If you had an opaque lid, you would only catch the respiration half of the equation. And so we didn't want to call it net ecosystem exchange because that kind of implies it's the whole ecosystem and would include the plants. But because these are focused just on the crust, you don't get that plant component. It's also different than just measuring respiration because the crust can photosynthesize. So we settled on this name for the carbon balance that we were looking at. So this zero line here would be no net difference. CO2 basically stays the same in the chamber over the course of the measurement. If they're above the line, it means the crust, the biocrust covered soil is emitting CO2, okay, due to various respiration processes, most likely. And then if it's below the line, like we can see here, and I've highlighted these particular 24-hour periods in green, then you're seeing photosynthesis taking place by the crust organisms. All right, and then I just have temperature down here on this line, and then I've shown rainfall events down here in the blue bars. Okay, and I'll go through these, and we'll just kind of follow these through the year because I think it's interesting to sort of take a really close look. One other thing, one of the technical challenges of working with this is, these systems, just like an eddy flux system, they always go down occasionally, and so if you want to get like a good estimate of the overall, integrated flux over time, you have to fill in the days that are missing with some kind of estimate of what they're doing. And so I worked quite a bit on trying to figure out a good way to fill in these gaps here. And there's obviously a lot of techniques for doing this in the literature. I found a relatively modern statistical technique to work really well applied to these chambers. And so here's I'm showing you guys a little bit of R code here for the R nerds out there. There's got to be some of them. But basically, what I'm showing is pretty straightforward. Basically, what I'm doing is I'm using this technique called random forests, which is a really excellent technique for finding predictive relationships between any set of input variables and an output variable of interest. Okay? And somebody wrote this really clever package that basically takes an entire data table that may have missing rows or it may have missing values in any of the rows, and it attempts to fill in every missing value in every different variable that you give it. Okay? And the way it does that is it sort of iteratively fits these random forest models. So it'll take the one that, for example, it'll take the column that's the most complete, and it'll try to predict the missing ones with all the other variables that are available. Okay, it'll do that for all the columns, and then it has a number. Then it redoes it again for anything that was missing. It eventually reaches some kind of stopping point, and it fills it in. So what I did was I fed it a whole bunch of different parameters in order to fill in those missing variables. And really, the script is much more complicated, mostly due to just managing the data and trying to get it in the right format. But after you do that, literally, you can fill in all your data with one line of code here. It takes it like several hours to run. But it's a relatively straightforward approach. What you feed it makes a big difference. And what I found was, in order to get these realistic values, I gave it, for example, I gave it all 5 control chambers here. Okay, and sometimes there were scenarios where one chamber wasn't working, but the other 4 were. Turns out, a good predictor of what that 5th chamber is just what the other 4 chambers are doing at that time, right? That makes sense. So those numbers I indicated those ones in purple here. Those I'm pretty confident that those are correct because they were always very strongly correlated, block 1 would be doing something, block 2 would be doing something slightly different, but the offset was similar. All right. There were also, of course, occasions when the entire system was down. That's shown in these red ones here. In order to help fill in those, I used a little bit of weather data that was from a nearby Castle Valley weather station when I didn't have weather data at the site. And also, I found, and this was kind of the secret sauce, if you put in lagged variables. So, for example, the flux from the day before is a great predictor of what that flux is doing at the exact same time the next day. Okay, and so once I put those in, I got very good at making fake data here. So yeah, and you can see, and I think something like this looks pretty good. You'll see some other examples where it does a good job. It also definitely misses things, and it does worse for the longer time periods. And so you can see here, here's a precipitation event. Probably there was some photosynthesis there, but the system was off, and the algorithm was not quite picking that up. So it's not perfect, but it does a pretty good job. Here's an example. So here's the next month, and you can see, it does a pretty good job like filling in these days. I think it's as good a technique as any for imputing the data and just making it so you can at least estimate a total yearly amount for the amount of carbon that's coming in or out. Okay, so now let's look at some of the trends that we're seeing here. All right, so we definitely see these daily cycles every day. All right, they're losing carbon sort of these are usually the middle of the day, and then down at night they come back down to around 0. This is sort of in the early part of the season. Okay, we see one example here where on this day where we get some precipitation, that you get substantial photosynthesis. The crusts have turned on and are fixing some carbon at that time. Let's see, February was more or less cold conditions, not a lot of precip, not a lot going on during that month. Okay, in March we have our first big storm here. Okay, and we see a substantial amount of photosynthesis taking place here. Oh, and by the way, I marked the darker green ones indicate days on which the net total carbon uptake was negative. So there was net uptake by the crust. These light green ones, there was photosynthesis, but if you average out the day, it's still above 0. So they're still losing a little bit of carbon from them. So this was one of the things I found the most interesting. Right here, where we have this big photosynthesis event occurring in March, I went back and I found a photograph from one of these days here, and this is what the chamber looked like. Okay, so really interesting. This was March 9th, 2006. It was one of these days right here. And so what you can see is that it looks to me like these crusts are turning on and photosynthesizing even under this, substantial bit of snow that has fallen on them. I would love to take the crust into the lab and prove 100% that that's what's going on here. But based on all the other numbers I've seen for the chamber, I see no reason to doubt that that's what's occurring here. And that's really interesting when we think of these as really hot systems. But it turns out for these crusts, some of the most important times for them might be when it snows. So I thought that was pretty cool. Okay, as we move further into the year, here's April, pretty dry. We start to see as spring hits, in addition to seeing these bigger daily fluxes in carbon loss, we also see that baseline level, those nighttime values, are actually off of the 0 line. And one thing we noticed here, when I first looked at the data, I was like, what is going on? The chambers are losing carbon practically all the time, except for those few wet periods where the crusts are photosynthesizing. Almost all the time, these crust-covered soils are giving off CO2. Okay, so and I was trying to think about how to explain that, and I'll talk about that after we finish going through here. Okay, let's see. So May, again, we still see this high baseline here, relatively dry. It's starting to get really hot here. The air temperatures in Moab are actually obviously quite hot in the summer, reaching up to 40 degrees C. And then in June, the baseline starts to come back down here. But we still have these relatively high daily fluctuations. And then this is interesting. In the summer, here's an example where we see a precipitation event right here. Instead of seeing the crusts turn on and gain carbon for themselves, you actually see them lose a whole bunch of carbon at this time. This is one of the biggest losses of the entire experiment was during this. And so, I was talking about at the beginning where they were doing that experiment where they were adding just a little bit of water, and the crust seemed to really hate it, and all the mosses died. You can actually see that same mechanism happening here in the control plots. At least one time during the summer, the mosses probably wetted up here. They did not get to stay on for long enough, and they ended up losing carbon. So it was a net loss for them. So you can see that effect occurring naturally. It just doesn't occur quite as often as they had done it in the experiment, enough to kill them, right? But if for whatever reason the precip patterns changed in the future, you could see these guys being sensitive and just dying off. It's not impossible. Okay, July, some more good examples of that same effect. Let's see. That was August. Okay, so September is starting to cool down. Here's a big precipitation event. This is probably rain. I mean, you can look at the temperatures; they're well above zero here, and the crusts are turning on and fixing at least a little bit during that time. Here's in the fall. The fall and winter is when we saw the most uptake in the crust. That's the time when they were most happy, at least during these couple of years that we were looking at them. It could vary. Obviously, the precipitation is quite variable year to year in these ecosystems. But here's an example where there's a bunch of precipitation, mostly rain, through October, and you see a lot of uptake in the crust during that time. But I think that overall, the total number of days where there was net uptake of CO2 was something like 12% across the entire study, and that's about the same number of days that the soils are wet in these ecosystems. So they pretty much turn on like clockwork when it gets wet. Okay, November starts to get into the winter. We see this is probably another example. I don't have a picture, but this is probably another example where we're seeing some snow. And again in December, a whole bunch of days where we're getting some precipitation. I don't know if this is rain or snow. Sometimes it snows there and just melts pretty quickly. It could be an event kind of like that. Okay, so question is, what's going on with these overall losses of CO2? Almost all the time we're looking at them, they're losing carbon instead of gaining it. I think, when these were set up, the idea was that there would be sort of a net balance. The average would be zero. But that's totally not exactly what we're observing here. And so these are the daily averages from those 24-hour periods over the course of the whole year. Actually, this is the course of the whole year and 9 months that we ran the chambers. Right? And so what you see is big daily average losses here in the springtime. There's a few days, those are those dark green days, when you have net uptake, particularly in the winter. But most of the time you're seeing big losses. Here's those summer days when you were losing a whole bunch due to those rain events. The annual total here was 62 grams of carbon per meter square of average CO2 loss from these biocrust-covered soils. And I'll put that in context here in a second. Okay, so if we look at this pattern over the course of the whole year, it's very instructive to compare it with what you would see from an eddy flux tower. All right? And an eddy flux tower is going to be integrating a large footprint of land that's going to include the plants. That's going to be the big difference there. All right? So the eddy flux tower is going to give you total carbon balance of the whole ecosystem, including the plants and the soils. All right? And if we look at the eddy flux values, we see an interesting trend. So this is an eddy flux tower that's I'm not exactly sure where it is, but it's pretty close to this site. It's not right at the site. But we think it should be probably pretty representative of the trends we'd see in that area. This is from another paper by Dave Bowling. And interestingly, in that spring period, he had like a dry year and a wet year. But in both of them, especially the wet year, you see a whole bunch of uptake ecosystem-wide, probably due to plant activity during this spring period here. Right? And interestingly, that's exactly the same time as we're seeing a whole bunch of loss of carbon from these biocrust-covered soils that are there. Right? And so I think that one obvious explanation, there's a few of them I'll talk about in a second, but one possibility here is that those chambers are not closed on the bottom. There could be plant roots that are growing down below them And as the plants are really active here in the spring, we're basically picking up a plant respiration signal at that time. That's one possibility. I don't have 100% the answer of why we see so much carbon loss from these soils all the time. Okay, let's see here. Okay, I wanted to put a little bit of context for those flux rates because, obviously this number 62 doesn't really mean much, in and of itself. But just to give you an idea, so if you look at eddy flux net ecosystem exchange amounts, so this is everything, plants and soils together, photosynthesis and respiration, they range, if you look at a bunch of there's a pretty good review paper I found that compiled a whole bunch of these together. They ranged from negative 100 to so uptake of 100 grams of carbon per meter squared to 150 grams carbon per meter squared loss. Okay, they're kind of all over the place, and I plotted them out here, and you can see some of them are gaining, some of them are losing, and this is kind of interesting. I mean, it gives you a good idea of the order of magnitude that you can expect for these numbers, at least if you believe the calculations from the eddy flux towers. I think it shows that those eddy flux footprints can be pretty variable depending on exactly where the tower is. You can't. I think we tend to think if this eddy flux tower is in this one spot, it's going to show exactly what the whole landscape is doing around that area. That may or may not be true, and seeing this big spread makes me a little bit suspicious of that. I think that probably some areas are gaining and some are losing, and it kind of supports that idea that these semi-arid systems are carbon's coming in and out, depending on the conditions and what's going on with the plants in that particular year. Okay, so this number, 62, it fits within this net ecosystem exchange amount of values here. It also sort of fits in what you would expect from just measuring respiration without the photosynthesis at all from the soils. Okay, and like I said, I think that a lot of that signal we're seeing of that carbon loss, once I thought about this, I was like, it's actually not that different. If we had just made the chambers opaque, we would miss out on that little bit of photosynthesis from the crust, but mostly we would just be measuring the ecosystem respiration. So that's kind of why I think this number sort of fits in this range of values from a variety of dryland respiration studies that we've seen out there. So so hopefully that helps to put that in context a little bit. Okay, so the question I have is, where exactly is all that CO2 coming from, from the soil? And I think we'd have to do some experiments to separate out exactly what the possibilities are. But here's kind of the range of possibilities that I've thought of. Number one, like I mentioned, there may be vascular plant roots that are growing down below this chamber. During that period in the spring when the plants are active, they are aspiring, and we're just seeing that CO2 signal coming up from those plant roots. Okay, it's possible. I think, to me, this is the most likely candidate, but it also, I'm not 100% convinced that that's all that's going on. Other possibilities could be that the biocrusts themselves are losing carbon over time. All right, that they're actually degenerating, and that the carbon in that upper layer is being reduced, and so they're contributing to some of that loss over time. I don't think that's you can't explain this whole 62 with that, because if you look at the amount of carbon in that biocrust layer, it's only a few hundred grams per meter squared. So they would be wiped out in 2 or 3 years if that was explaining all of it. Okay, there's more carbon down below the biocrust as well, and so we could be seeing a little bit of signal of reduction in the soil organic matter over time just within these plots. And of course, it could be a combination of these things. The kind of wild card reason, there is a thick, pedogenic carbonate layer at this site. It makes it really hard to dig into the soil there. It's not far below the surface. Obviously, carbonates, can produce CO2. It is possible. I don't think the patterns we see in the timing of when we're seeing the CO2 loss suggest that the carbonates are the big source of this. But it's hard to know for sure that they're not contributing at all, unless you do some isotopic studies to try to separate out that carbonate contribution there. And I've seen there's varying reports in the literature. There's some papers that say, yes, the carbonates can be important. There's others that say that they are not. I know that Sasha Reed asked Bill Schlesinger, who's worked a lot in these systems, about that. And his take was that the stoichiometry doesn't work out at all. There's no way it could be. There's just not enough. It has something to do with the chemistry. There's not enough of part of the equation to make it give off enough CO2 over time. So anyway, I think that remains to be tested. I would love to explore this more, but we'll have to get together the funds and everything to be able to actually do that. Okay, so now what about those warming lamps? Okay, what effect did they have? That was the main purpose of the experiment, was to figure out climate change effects on the carbon losses here. Okay, what kind of effects did it have? All right. So what I've done here is I've graphed the cumulative carbon balance, from these chambers over time. And what I've done is I've separated out each chamber individually. Okay, so you can sort of see the variation among them. Okay, which I think is important to do because it sort of paints the picture of we're not exactly sure what's going on here because they overlap with each other quite a bit. So we have the warmed ones in red here. They lose a lot more carbon here in the spring, just as we saw with that annual rate. They lose less over the winter here, and then again in the spring. If we look at the first full year of data, we see that on average the warmed plots are higher, but there's a huge amount of uncertainty there. My estimate here is they're 20% higher, plus or minus 60%. Okay, so it's hard to say exactly what's going on there because they overlap quite a bit. We have these three that seem to be higher, but then there's these two that are kind of mixed in with the controls. The controls are all over the place. And this is not uncommon when you're doing these types of measurements on the soil. I guess maybe we should have had 100 auto chambers instead of 20 of them if we wanted to constrain that number down to a better level. It's hard to do. It's hard to do. But this, for an experiment like this is all we have. So I feel like you have to look really carefully at all 5 of the chambers and see what can we conclude here. In a way, it's more likely that the warmed ones are losing some, but we just can't be particularly certain about that conclusion. Now, in contrast, if we look at specific times of the year, there's a couple of times when there is a really clear treatment difference. Okay, so if we had separated out certain aspect or certain portions of the timing here. Okay, and the main time that we see differences are essentially when the biocrusts are active. Okay, so when they're wet. So here, for example, is the days, those green days from the time course where photosynthesis was active. All right, during those days, we see much greater carbon loss in the warmed plots than we do in the control plots. Okay, and these are substantial extra losses from these crusts. That drives these seasonal trends as well. These daytime seasonal trends, at least in the winter and the fall here as well. So we do see some treatment effects, and it seems like when the crusts are active, the heating treatment may be a little bit unfavorable to them. It's negatively impacting their ability to take in that extra carbon. Okay, and then finally here, there's some really interesting relationships that we see if we look at all the different days that we measured. I mean, because we have a huge amount of data. There's something like 15,000 data points over that year and 9-month period. And if we look at all those together and compare them to environmental variables, we see some pretty interesting trends suggesting that the crusts are really responding, or these crust-covered soils can be relatively easily predicted. What their rate of carbon loss is going to be. Okay, so here's an example. I have a bunch of these in the paper, but I'll just show you guys a couple of them. Here's a correlation with air temperature, and then here's the total carbon balance here. As it gets hotter and hotter, they tend to lose quite a bit more carbon. Those really hot days, they're losing the most in the summer. Okay, and you can also kind of see the days on which photosynthesis is occurring. They like these much cooler days right here. And then I checked these to make sure these are the days when we were seeing that subnivean photosynthesis occurring. These are all the values that we're looking at. These are only from when the soils are wet. Okay, you see a different trend when they're dry. This is in the paper if you want to check it out. When the soils are wet at night, you see pretty much exactly what you would expect. Without photosynthesis dragging those values down, you just see this kind of exponential relationships here with a Q10 that's pretty much in line with what you would expect for a respiration process in these types of soils. Okay, all right, we're back. Okay, yeah, this is kind of like one of the last things I wanted to show here. But So if you look at the daily time course, we have so many days that we measured. You can kind of get a picture of how those daily cycles are consistent over time. And you can see that the peak of respiration typically occurred sometime around 1:30 p.m. And by the way, I think that it doesn't necessarily indicate that that's when the microbes are the most active. That just indicates when the CO2 is coming out of the soil. All right, and there could definitely be some physical factors there where it heats up in the middle of the day. It might, sort of cause the soil to expand and push air out of there at that time of day. And I think there's probably, in order to explain those daily cycles, you need a little bit of, you've got to consider those physical factors as well as, biologically when the CO2 is being respired. And then for photosynthesis, if we're on these days when there was photosynthesis, it would peak around 11 o'clock. It's kind of what I would expect. The plants would turn on in the morning, and then it would start to get hotter and hotter as the day goes on, and they might, downregulate a little bit at that time. But yeah, so that was kind of what we saw with that. Okay, so a couple of conclusions. Overall, our crust photosynthesis was clearly measurable. Right? We could definitely tell when they were photosynthesizing, even under snow, which was kind of neat. But I didn't think that those total photosynthetic rates were particularly large compared with the background CO2 losses that we were seeing all the time. And I'm going to guess that's because the plants are doing way more photosynthesis than the crusts. At least, there's some tricky scaling things to work out, so I can't say that 100%. But it looks to me like the crust photosynthesis, it definitely occurs, but it's maybe not like the hugest component of the photosynthesis in the ecosystem. Okay, and then as we saw in the middle of the summer there, that mechanism for the moss death that Sasha's paper had documented pretty clearly, we could see it happening in the chambers when they got a little sprinkle of rain during hot conditions, but it just didn't seem to occur as often as you would need in order to kill the mosses like they saw. And then I'd say, all in all, our data suggests that there's definitely a risk that with these higher future temperatures, that the crusts are going to, especially because we saw when they were active, they didn't really like the warming lamp treatment. It negatively impacted their carbon balance, which is probably ultimately bad for them. So, I think we can say provisionally there's some evidence that warmer conditions are probably not going to be great for these crusts. And then, finally, is next steps. I think I'd love to get a better mechanistic understanding of exactly which of those different flux pathways was contributing to the carbon loss throughout the ecosystem. Maybe it'd be great to do some experiments where we isolate, the crust and then the subsoil, and then just try to pin down the carbonate thing to make sure that's not driving the trends there. So, okay, yeah. So that's it. Thank you guys very much.
So you mentioned the mosses as a big component of the crust.
Yeah.
Are there also, I'm assuming there are also crusts consisting of cyanobacteria and other algae associated with the fungi. Is that true in this case?
Yeah, in this case, and these crusts have.
What percentage might you see of those?
I don't. As far as the biomass that we made up by the mosses and these crusts had lichens. They probably had cyanobacteria. And almost all the crusts have some cyanobacteria in them. They probably have all those other components. As far as the biomass, I don't know. Jane probably would have an idea of that, but I'm not 100% sure about that.
Predominantly moss?
I kind of feel, I'm guessing that the mosses were driving the photosynthesis trends in these ones, but I don't know that for sure. Probably the lichens were contributing a little bit as well, because there's quite a bit. That black stuff on top is a type of a lichen, so that's probably contributing. Yeah. Yep.
So I think you've got root production. So if your NPP is 150 grams per meter squared, your root production is probably 90. But I'd say a big part of that's going to be rise of deposition. You probably have 10, 20, maybe 30 grams of rise of deposition per meter squared.
Totally.
A big part of that.
Yeah.
That soil respiration.
Right. Yeah. If there's shrub or there's grasses and forbs here too. If they're spitting sugars out below our chamber somewhere that we can't see, and then the microbes are respiring that, yeah, I think that would be a good candidate. Yeah.
Well, along those same
Yeah.
lines, didn't you hint that maybe yesterday when we talked that there might be some fungal connections directly from roots into the
Mhm.
crust?
Yeah, and I think
Transporting carbon directly to the fungi into
Right.
the firing.
yeah, no, it's possible. I think no, these chambers they go down 20, 30 centimeters. So theoretically, that's going to cut off any fungal connections to the nearby stuff, unless the fungi are going way down, which who knows, maybe they are. But yeah, yeah, yeah.
You kind of touched on why increased water treatment holds a lot of costs.
Mhm.
But I was wondering if you could explain that a little bit more. Just
Sure.
is that if there's more water, it then when it's really warm out,
Yeah.
it dry up. Is that due to the rate that they're drying up?
Yeah, exactly. That's what's happening. I think what's happening is maybe I didn't explain it super clearly, but you sprinkle a little bit of water on them, you'll see them green up almost immediately. All right, but it requires a lot of energy for them when they're in that desiccated state. It requires them to they save up sugar. In fact, if you take like a moss and you can measure it, a substantial portion of their dried biomass is sucrose. And they save it as sugar in there. They use a bunch of those energy reserves to rehydrate and restart all of the photosynthetic machinery that's in the moss. They then need to photosynthesize for a while to make up for those reserves that they just spent rehydrating. If they don't have enough time to do that, then they're going to slowly lose those sugar reserves over time, and then eventually they'll bite it. Basically, that's what they saw. So yeah. Yeah, Kendall. Yeah, go for it. Yeah.
So the figure where you were looking at the wet during the daytime and then the wet during the dark.
Yeah.
I was curious. It looked like there was some photosynthesis happening when it was dark, and I was curious.
Oh yeah. No, that's a really good point. Right. I mean, theoretically, in the dark here, we should not be seeing anything below zero. I agree with that. I think these ones down here, probably some kind of anomalous value, probably some kind of error. Okay. These ones that are like really close to zero, I think that's more just kind of like the noise in the system a little bit. But no, I do not think that they are taking up CO2 in the dark. Yeah. It's a good observation, though. Yeah. Yeah, Andrew.
I had a question about your random forest and fertilization. Is there a way when you're doing that to sort of bootstrap the data that you do have to see how well that technique can predict actual data rather than just gaps?
Yeah, you could do that. I didn't do that. But yeah, you could purposely eliminate a bunch of data, X% of your data, then run the algorithm, and then see how well it does at predicting the known values. Yeah, absolutely, you could do that. It would be a good thing to do. It was just already kind of involved, and so I didn't quite go that step, but it's a good idea as a check. Yeah, I think you'd find that it would do pretty well. I mean, because like the correlations are really good. I mean, it could predict, in many cases, 85, 90% of the variation in there. But I didn't test it on the totally unknown ones, like you're saying. Yeah. Yeah.
Do you think that there might be a small amount of carbon fixation that's not driven by photosynthesis?
Interesting. Like some kind of
Like
chemoautotroph. Is that the right
Yeah.
one? Yeah. I guess it's possible. I mean, who knows what's in there and what kind of metabolic capabilities they have there. I've never seen anything about that, but yeah, I wouldn't rule something. It would probably be very small, but
yeah, it would
Yeah.
be small, I'm sure.
yeah. You'd kind of. I mean, those guys might get outcompeted by the ones who can do photosynthesis, which is much more efficient, but
Well, I mean, you could
energetically.
have nitrification as an example.
Yeah. Yeah. No, it's a cool idea. Yeah, it could contribute a little bit. Yeah.
On those lines, I think
Yeah.
you could look in Jeff Barrett. He worked down in Antarctica, and they
Oh,
were
okay.
doing carbon flux on these really carbon poor soils.
Yeah.
And they found there's a lot of physical and chemical reactions were dominating carbon flux.
That's
So
really
I think,
interesting.
I
Yeah.
don't know if they had carbonates, and I don't know if they totally figured out what was happening, but they did some sterilized plots and found they had obvious carbon cycling that they're picking up in abiotic plots. So.
Totally. Yeah, I mean, I'd love to do that partitioning exercise. I think if you ruled out all the other things first and you still saw things like that, then you would know if that was occurring. And I'd be super interested to find out if that was the case. Yeah. Yeah. Okay. All right. Well, yeah. Thank you guys so much.